CRISPR can be used on humans, but has it? We need to consider the ethics associated with doing such a thing, particularly in engineering a baby, as the changes will be passed on to their offspring. Plus, what about if the process goes wrong? Well, it's been done. In this case, the target was a gene called CCR5, which makes the chemokine receptor type 5 protein, a G protein coupled receptor. So why was this done? Well, the receptor is a point of entry into cells for HIV and Black Death, and a father who wanted children and who had HIV didn't want to pass on the virus to his children. So the thinking was, if the receptor was mutated or its expression stopped, then the children wouldn't be infected with HIV. Sound idea. Now, CCR5 is not just a disease receptor, it is involved with the immune system. So changing the receptor's function could also impact the way the immune system works. Now, the CRISPR approach was used to try and mimic a natural mutation that occurs in 1% of Northern Europeans, where they have lost 32 bases from both copies of CCR5. This mutation results in the truncation of the protein, which means the protein does not reach the cell surface. In the two CRISPR babies, one has lost 15 bases in one CCR5 gene and none in the other, and the other baby has gained bases in one CCR gene and lost bases in the other. So neither baby has the Northern European mutation. So what does this mean for the babies? Well, they might die young, as it is known that Europeans that carry the natural mutation are 20% less likely to reach the age of 76, and they might be more susceptible to other infections. There is also the problem of off-target effects to be considered, which is when the CRISPR guide RNA recognises other targets in the genome and carries out off-target edits. The 3 scientists involved in producing the CRISPR babies have received fines and were imprisoned. Okay, so let's briefly look at a couple of legal uses of CRISPR to treat humans, as I wouldn't want you to think it is all bad. In 2020, phase 1 clinical trials started using CRISPR to treat testicular cancer that was resistant to the usual treatments. In the procedure, T lymphocytes were collected from patients and CRISPR was used to disrupt 3 genes involved in T cell targeting. A lentivirus was then used to introduce a new targeting system that would effectively point the T cells at the proteins specific to the patient's cancer. The new T cells were then transfused back into the patient. 2020 also saw the first use of CRISPR gene therapy administered directly into patients' bodies. In this case, it was to treat a leading cause of blindness in children, LAC10, caused by mutation in the CEP290 gene. CEP290 seems to be involved in retinal development, and some 35 mutations in the gene have been found. How the gene regulates the development of the retina is unclear. For the treatment of LAC10, CRISPR is loaded into adeno-associated viruses and injected into the eye. CRISPR then deletes a mutation in the CEP290 gene that causes LAC10. Okay, so that is 2 promising and legal uses of CRISPR to treat patients. Finally, and very quickly, I must just mention CRISPR and climate change. Basically, you name it and someone is trying something using CRISPR to change organisms to help reduce climate change or change organisms to withstand climate change. In the last few years, I've come across projects on helping protect coral reefs against temperature and pH changes, cows and methane production, making livestock more disease resistant, producing drought resistant plants, and many, many more. However, and this was covered earlier, there is a problem in that a lot of the issues we face are not one gene wonders and may involve the need to change many genes, and we need to identify all the genes we need to alter.